Hey, this is Mrs. Reichelt. In this next video, we're going to go ahead and talk about blood groups and transfusions. So basically, when you lose a large amount of blood, it can cause weakness. In many cases, or in some cases, and maybe I should say, it'll call for a transfusion. So a transfusion is the only real way that you can replace blood very quickly. So in this case, if you lose between 15 and 30% of your blood, it will cause weakness. If you lose over 30% of your blood, you can go into shock. When you go into shock, that can be fatal. Luckily, there's transfusions, which is a way to replace blood very quickly. That means that you're getting blood from a foreign material, so another person maybe donates blood and you take that. If you don't have a transfusion, the only way to replace blood naturally is going to be through hematopoiesis. Transfused blood must be of the same group. So that means if you have type A blood, you need to get blood from type A in most cases. Human blood groups are going to be genetically determined. So you have antigens, which is substances that your body recognizes as being foreign. When I think of an antigen, I usually think of a pathogen. So a pathogen is a foreign invader. Antibodies are what your body recognizes being self or body cells. Antibodies usually are what attacks the antigens or the pathogens. Then blood is going to be typed by using antibodies that will cause blood with certain proteins to clump or agglutinate. So if the proteins that your blood is reacting with, they don't know them as self antigens. If they're self antigens, they won't attack them. If they're non-self antigens, then your immune system will attack those blood cells. In the human body, there are about 30 different types of red blood cell antigens. However, the most vigorous transfusion reactions are caused by ABO and Rh group antigens, and we'll talk about what those mean right now. So basically, blood is going to be typed based on the presence or absence of two antigens. If you have the type A antigen, then you're type A. Your genotype could be IAIA, which would give you type A blood, or you can be heterozygous for it, which is IAIO. That would also give you type A blood. If you're type B blood, you could be IB or IBIO. If you do not have those antigens, then you are type O blood. So that means that you would be IOIO. When we talk about antigens in this context, we're talking about self-antigens. So a self-antigen is something that your body recognizes being itself. So an antigen on its own would be a pathogen, something that your body recognizes as being foreign. But in blood grouping, we talk about self-antigens. If you have both of those antigens, then your blood type is AB. So the genotype would be IAIB. So the presence of self-antigen A gives you A blood. The presence of antigen B gives you B blood. And if you don't have either of those, you lack them, then your blood type is O. So hopefully that's pretty easy in here. When you're getting a blood transfusion, if you have type AB blood, that means that you can receive blood from A, B, AB, and O. We would call that type the universal recipient. If you have type B blood, you can only receive blood from B and O. Same with A, except for instead of B, it'll be A. And then blood type O, we call the universal donor. That means that it can give blood to A, B, and AB, but it can only accept blood from type O. In addition to the A, B, and O groups, we also have the Rh blood group. This is what gives your blood positive or negative. So you could be A, B, or O, and you could be then positive or negative as well. So you can be A positive or A negative. Most Americans are Rh positive. It's the dominant trait. So that means that you can be positive positive, positive negative. That would be heterozygous, and that would give you a positive outcome. Or you could be positive or negative negative, which would give you negative blood. Problems can sometimes occur when you mix positive and negative blood together. If a female is pregnant, so I'm going to draw a female. So if a mother is pregnant, and we'll say that this circle right here is going to be her fetus. If the mother has, let's say, a negative blood type and the fetus has a positive blood type, if the fetus's blood gets intermixed with the circulatory system of the mother's immune system will produce antibodies that will attack this foreign blood, even if it's a fetus. So what we do instead of having that occur is a female, if they have a negative blood type, they will get a shot of RhoGAM. This RhoGAM shot basically tells this negative blooded female, if you come into contact with positive blood, that's okay. Don't attack it. It will make it so the mother's antibodies or immune system will not produce antibodies against that positive blooded fetus. If the mother does not receive that RhoGAM shot and they have a negative blood type, the first pregnancy will go typically without problems. However, after that baby is born, the immune system will be sensitized. What that means is the immune system will know that something shouldn't have been there, and now that'll be basically, if it comes into contact with that again, it'll produce antibodies. In the second pregnancy, the mother's immune system produces antibodies that attack the Rh positive blood, which can cause hemolytic disease for the newborn. So if a pregnant female has let's say a positive blood type, and the infant has a negative blood type, the mother's immune system doesn't care if that fetus is negative. Just like if you were a positive blood type and you needed a blood transfusion from a negative blood type, your body would be okay with that. The difference is if you are a negative blood type and you receive blood from a positive blood type, your body will reject that positive blood, just like if you're a female who is pregnant with negative blood type and the fetus has a positive blood type, it is possible for the mother's immune system to attack that blood. All right, so basically we're able to blood type a couple of different ways. When you blood type, we can't do that in class because it's an online class, but if you're given a blood typing lab, you're basically going to have a card that has four different circles on it with chemicals on them. Basically, there's going to be one that determines A blood, one that does B, one that does the positive and negative, and then one that is the control. We're looking for something called agglutination. If agglutination occurs, that means that that is your blood type. So I'll show you what that means right here. If you wanted to see if you were type AB, you would have agglutination, which is this. If it looks grainy, that's called agglutination. So if you agglutinate for A and B, that means that you have type AB blood. If you agglutinate for B only, that means that you're type B. If you agglutinate for type A, that means you're A. If nothing happens and it just kind of looks like the control, that means that you're type O. The same thing will occur with the Rh. So that's basically wraps up blood transfusion and blood typing. I hope you found that helpful.